Started at 6:30, so we want to get done before midnight. And welcome. We are glad you're here. Let's start with prayer. This is a prayer for all of our students to God, but especially our seniors. So in the name of the Father, Son, and Holy Spirit. O good and gracious God, I have knowledge, so will you show me now how to use it wisely and find a way somehow to make the world I live in a little better place and make life with its problems a little bit easier to face. Grant me the faith and courage and put purpose in my days and show me how to serve in the most effective ways. So my education, my knowledge, and my skill may find their true fulfillment as I learn to do thy will. And may I ever be aware in everything I do that knowledge comes from learning and wisdom comes from you. We ask this in your name, O Lord. Amen. Father, Son, Holy Spirit, amen. Saint Robert Bellarmine. Thank you. Miss Nuno is going to share our land acknowledgement statement with us, and then I'll come back.
Every community owes its existence and vitality to generations from around the world who continued to make the history that led to this moment. We recognize that all of us at Bellarmine learn, live, and work on or near the unceded territory of the Coast Salish people. Our campus is situated on the traditional lands of the Puyallup tribe. As we gather, we also want to acknowledge the lands of the Muckleshoot, Nisqually, Squamish, Snohomish, Sammamish, Duwamish, and all traditional lands on which we live and work. We want to acknowledge the histories of dispossession and forced removal and reflect on this history in ways that honor the memory of those dispossessed and keep us committed to the cause of social justice for Indigenous populations.
So again, thank you for being here this evening. As it turns out, this is our 40th anniversary for the Marine Chem program. So yeah, shout out. And these are some of the folks, I think I might have shared this last year, but these are some of the folks throughout those 40 years that have been involved with our program. I think over 1 million people have been through this program, probably, maybe 2, something like that. I actually just got my insurance agent was telling me about his upcoming ankle replacement. Who's going to do it? One of our Marine Chem graduates from 2005, of course. So they're out there doing lots of good things, and they're in here doing lots of good things. But this wouldn't be possible without one of our greatest leaders here at Bellarmine, and my mentor, personally, Ron Nielsen. I don't think Ron's here, but he is still mentoring a few of our students. He retired last year, and he is going to be mentoring a few next year, despite being 120. So it means just keep working hard. Okay. Our format tonight, we have 7 presentations. Each presentation is going to be about 12 minutes. We reserve about 6 minutes for questions. Again, make them tough. And each student is going to be presenting the next student in line. After the 4th presentation, we'll take a break. Out here is, and some people have already found it, I hope they're left, there's some cookies and some drinks, water, and make sure that you check out the posters too. And lastly, I didn't really see people grabbing the commemorative wristbands. So grab the commemorative wristbands on your way in, out, and if you miss it this week, we'll get it for you next week. So with that said, I'm going to sign, one more thing. We've been practicing, and all the practice has gone perfect, and then we may have a few issues here tonight, so bear with that. Students may need to get in and out of their notes, but they are all prepped and ready to go. So. With that said, am I off? Yes. Our first presentation is going to be Nate Lilly.
Crabs. They're not just for dinner, they surround us. They thrive in our ecosystems and they support our ecosystems. Hi, my name is Nathan Lilly. I appreciate you all taking the time out of your Wednesday night to join us. And my project is the microphotography and DNA sequencing and identifying Puget Sound crabs in the Megalope and Zoe stages. Let's begin with why are crabs important. Crabs are vital to marine and freshwater ecosystems as both predators and prey, contributing to nutrient cycling and maintaining habitat structure. They are also an important food source for many human communities and a key part of the fishing and aquaculture industries. European green rock crabs are an invasive species that appeared in 1998 in Washington State, but did not become relevant until 2018 when they had a massive species bloom. They are voracious predators that decimate native shellfish and surrounding ecosystems. This pushes us towards my background research. From Whale, Scholes, Richter, et al., the zoeal development stage is after hatching, the larval, which is the zoe, look like shrimp and rely on currents and appendages for movement. They undergo molts, where swimming appendages reduce, walking legs form, and the number of zoeal stages varies by species. Jean et al. Covers the megalope stage. This is the final and largest planktonic stage of decapod crabs. At this point, they start to resemble small crabs, show walking legs, and longer abdomen. The crab transition is the megalope stage serves as a bridge between the larvae and the juvenile crab. It is a critical step where crabs prepare to leave the plankton and begin life on the ocean floor. The Washington Department of Fish and Wildlife have created an identification key which is limited to the juvenile and adult stages of Puget Sound crabs. This pushes us towards previous Bellarmine studies. McKinley Barney, who graduated in 2025, and Taylor Flattery and Alec Woodruff in 2016 both completed studies that collected Puget Sound crabs in the megalope stage using fluorescent light traps. After collection, they were photographed, and these images were used to gather data about physical characteristics across several different species. These measurements were then used to create identification keys for use by the public. On the screen, you will see identification keys that were created from the 2016 study. This pushes us towards our problem statement. Previous studies do not utilize microphotography adequately enough to distinguish different species of Puget Sound crab by their physical characteristics. Identification keys that have been made in the past are not complete in their analysis of the sample and do not include the megalope and zoe stages, or simply are not made available to the public. More work on proper identification is necessary. DNA extraction and genetic sequencing in this process has not been completed ever at Bellarmine before. My intended audience is the public, such as the general public and commercial fishermen. They can use my study to identify crabs that they find in the wild in younger stages, as well as scientific research groups and professors, like groups from Seattle University, University of Washington Tacoma, and the Washington Department of Fish and Wildlife, using my procedures for physical identification as well as genetic identification. My objectives of my study is to collect samples of Puget Sound crab during the megalope and zoe stages, update previous methods of microphotography to improve details focus, use microphotography to gather images of general and specific characteristics of each crab, and utilize DNA procedures to gather genetic information to differentiate species. My final objective is to create an identification key that uses physical characteristics combined with DNA to distinguish species of Puget Sound crab. An overview of my materials and methods includes the light trap preparation and setting the trap, taking the samples from the trap and analyzing them at the Bellarmine Marine Chem Lab for DNA and physical characteristics, as well as the DNA setup and its procedures. Moving on to my crab collection. Sampling occurred weekly at a fixed location, which was Day Island Marina, and the light trap system, which is pictured on the top right of the screen, included a 15-volt battery for illumination, a 500 micronewton mesh container to prevent samples from slipping through, and the trap was lowered by hand once a week, approximately 3.5 feet below the surface. The remaining line was wrapped around a fixed fire department water resource on the dock. The trap was designed to attract and collect small marine organisms overnight. The mesh size allowed water flow but prevented the loss of specimens. After collection, the specimens were taken back to the Marine Chem Lab for observation and analysis. Once at the lab, the samples were processed with forceps, which were used to carefully separate organisms from other debris. The selected samples were then placed in a 1,000 milliliter container. These containers were stored at a controlled tank at 50 degrees Fahrenheit to maintain environmental consistency until microphotography was performed. Each crab was imaged individually. The crab was positioned on a millimeter scale to allow precise morphological measurements. The microscope used a 2x ocular zoom, and an iPhone 16, also at 2x zoom, gathered high-resolution images. 4 standardized photos were taken of each crab: the whole body, one of the antennae, one of the dorsal spike, and one of the abdomen. Pictures were also taken of the crab while it was alive as well as preserved. The picture on the left-hand side of the screen shows the alive crab, and the right-hand side of the screen shows the preserved version. Speaking of preservation of samples, the crabs were placed in 1 milliliter tubes, which were then added 0.5 milliliters of 95% ethanol, and the tubes were labeled with the date as well as the abdomen length, so I could differentiate species without knowing the genetics yet. These tubes were stored at 38 degrees Fahrenheit for long-term preservation. Moving on to the DNA portion of my methods. For an overview of DNA extraction, first cells are broken open to release their DNA. Chemicals are then used to remove proteins, membranes, and enzymes that could interfere with PCR or damage the DNA. This purified DNA is used as a template for PCR, where specific DNA sequences are amplified millions of times for analysis. For this portion, I used WSU's extraction methods, as you can see on the screen up top. The second stage is the PCR. PCR is where a DNA sample is placed in a thermocycler, which is a machine that rapidly changes temperature. First, the DNA is heated so that the 2 strands separate, which is called the denaturation stage. Next, the temperature is lowered so that DNA primers attach to the target sequence, which is called annealing. Finally, the temperature is raised slightly so that the DNA polymerase enzyme builds new DNA strands, which is the extension. The thermocycler repeats these steps many times, doubling the amount of DNA each cycle, which produces enough DNA to be easily studied or analyzed. Our denaturation stage was set at 97 degrees Celsius for 25 seconds. The annealing stage was set at 55 degrees Celsius for 35 seconds, and the extension stage was set at 72 seconds. These procedures were ran for 35 cycles, which amounted to 156 minutes, which is about 2.5 hours. The primers in this study were changed to the JG-LCO, which is a forward primer, and the JG-HCO, which is the reverse primer, targeting the cytochrome C oxidase 1 gene. This was due to the fact that these primers targeted invertebrate animals. The final stage of DNA was the gel electrophoresis, which is a method used to separate DNA fragments so they can be seen and analyzed clearly. DNA samples were placed in a gel using a pipette after the green dye was added, and the electric current was applied. Because the DNA has an a negative charge, it moves through the gel towards the positive end. Smaller DNA fragments move faster and travel further than the larger ones. For my physical results, as you can see on the screen, there's 3 images of the crab. The far left indicates the photo that specifically targets the antennae. The middle targets the abdomen, and on the far right, the photo targets the dorsal spike. On the top right shows a data table which indicates how I mark the measurements for each crab. Sadly, my DNA results were inconclusive. As you can see, my electrophoresis was slightly unsuccessful. The control is seen on the left-hand side of the screen, where the base pairs have separated, but 4 out of my 5 DNA samples ran together, and one other one did not run at all. The DNA was not conclusive or complete enough to be cleansed and prepared to be sent to the laboratory for genetic identification. My discussion and conclusion includes my identification key. Since there was no DNA, I was only able to identify 2 species for sure. On the left-hand side of the screen is the purple shore crab with an antennae at 0.65 millimeters, eyes protruding horizontally, a back spike at 1.2 millimeters, and a rectangular carapace, which is basically the body of the crab. The abdomen is also at 3.2 millimeters. The red rock crab on the right-hand side of the screen has an antennae at 0.91 millimeters, rounded eyes that are closer to the midline of the crab, and a back spike at 1.39, an oval-shaped carapace, and an abdomen length of 1.94. Some of my limitations in this study was that the previous DNA was lost. I originally did the 3 stages of the DNA process at Seattle University, and they were placed in the marine chemistry fridge. But 1 month after they had been preserved, I went back to check on them, and they were missing. I was also restricted from my sample location after about 1.5 years of research. I was removed from the dock by the dock manager, and upon asking him for a reason, he did not give me one. He just asked me to leave. Another limitation inside of my study was that crabs would curl up after preservation. After sitting in ethanol for about 24 hours and sitting in the fridge, they would curl up like a dead spider. The legs would retract, the abdomen would retract, and the head of the crab would move towards the body. For future applications for my study, commercial fishermen and the general public can use my study to identify crabs in the smaller developing stages, as well as marine chemistry students. Marine chemistry students can improve my DNA methods and hopefully get better results, and improve my photography methods as well as physical characteristics. Researchers from UW-Tacoma and Seattle University are also completing similar studies on the megalope stage, so they could also use my physical characteristic procedures to help them. These are my references. And I would like to extend a massive thank you to Mr. Nielsen, who originally got me a permit to even get this project started, allowing me to sample crabs from Puget Sound. Mr. DeGroat and Mrs. Etter for pushing me to get all the little details of this presentation done. Professor Holtgren at Seattle University for showing me how and why to do all the DNA stuff. And my family for listening to numerous presentation practices. And finally, the Marine Chem class of 2026. I'm looking down at all of you, and all I see is future greatness. You guys are amazing. Thank you. Questions? Yeah.
Why did you choose ethanol as the preservative?
So the question was, why did I choose ethanol as my preservative? To be honest, I believe it was the highest alcohol-based preservative we had in the Marine Chem lab. I could be wrong, but that was also what was provided to me by Mr. DeGroat. Sorry, I'm not trying to put you in the spotlight. But.
Alcohol's a nice drying agent.
Alcohol's a nice drying agent, yeah. Yes.
So you collected samples a couple of times. How many crabs were you getting in each sample?
Yeah, so the question was how many crabs was I getting each time? So every time I sampled, which was once a week for about 1.5 years, I would get anywhere between 0 and 150 crabs every time. So it was microphotography of every single one of those. Yeah.
Do you think it's a reason why the DNA analysis didn't work out?
So the question was, I knew this one was coming, why the DNA did not work out? I do not know. This was the first time these DNA processes had been run at Bellarmine. I'm working with Professor Holtgren, going through my methods and everything I did to see where I possibly made an error, but nothing so far. Not sure.
Did you only run DNA on the preserved samples or any other fresh?
Sorry. The question was, did I run any of the DNA on the live samples? So to run them, I have to crush them into little microscopic pieces. So they kind of have to be, oh, do you mean like?
Before you preserve them.
Right. No, I did not. Just for the reason that for time. So I couldn't, if I did that, I would be running it once a week for 1 year. And we don't have the supplies here for that. What was my favorite part of the project? To be honest, I think it was, so when I went down to the docks every time to go get samples, I had to have at least one other person with me per the permit. So I think it was making some of my friends go down there at 10 p.m. because I forgot to set the trap that night, or it was just going down with my family, spending quality time with them. All right, thank you. right. And next up, we have Aaron Barnhart with evaluating the use of intravenous acetaminophen in pain control by EMS transports.
All right, hello everybody. My name is Aaron Barnhart, and I'm here to present to you evaluating the efficacy of intravenous acetaminophen, otherwise known as IV Tylenol, in EMS transports. Why did I choose IV acetaminophen, which I will refer to as IV Tylenol for ease in the study? I chose this because opioids present a high risk within our society, and there were 3,100 opioid deaths in Washington, which has created a need for non-opioid pain management alternatives. And I chose IV Tylenol to find if it was a potentially safer alternative than opioids. What is IV Tylenol? It's a non-opioid analgesic, which means it's a painkiller that does not bind to the opioid receptors in the brain. How it works is it gets into the central nervous system by bypassing the first metabolism in the liver, and is quicker than oral Tylenol by 10 to 30 minutes because it does not have to be digested. Another benefit is it does not have to be taken orally, so if a patient is vomiting, it can go in without having to be thrown up or stomach. Usages are post-anesthesia, after surgeries where patients need pain killing, and then pain relief in general, such as EMS transports, which is what I'm studying. So IV Tylenol, in comparison to opioid, has many ways to compare them. First are the contraindications or reasons you can't take the IV Tylenol or opioids. For contraindications for Tylenol, there's allergies or severe liver disease because it goes through the liver. And then contraindications for opioids are allergies, pregnancy and breastfeeding, and then substance use disorders. Side effects, IV Tylenol has virtually zero side effects, but opioids tend to have a lot of side effects, such as nausea and vomiting, risk of dependence, and then others shown on the chart, such as the immediate and long terms shown. The logistics also play a large part, and something that surprised me when I looked into these two pain method managements were the extensive ways that opioids had to be stored. When hearing from my mentor in the ambulances, I heard that IV Tylenol has to be in a lockbox within a safe, and then it's also a controlled substance, so two people need to be there to extract the opioid, so it's a lot of logistic concerns when administering. And then safety is also a concern, as losing opioids can be a lot more detrimental than losing IV Tylenol. When looking at previous studies, I found a lot of limited and mixed data. The two studies, Singla and McNichol et al., found positive results, saying IV Tylenol has been proven effective in orthopedic surgery and lower risk for adverse effects post-procedure. However, I did find some negative/neutral data with Marcotte et al., citing that IV Tylenol offered a limited benefit in colectomy patients, which is a type of colon surgery. These lead me to my problem statements. First, I wondered that there is a gap in knowledge regarding if IV Tylenol is an effective pain management alternative to opioids in EMS transports. And second, it is unknown if IV Tylenol has different efficacies for different types of pain. I hypothesized that given previous studies about the efficacy of IV Tylenol in procedures requiring anesthesia, I hypothesized that IV Tylenol will be statistically effective in reducing pain scale scores in EMS transports. Secondly, I hypothesized that IV Tylenol will have a similar pain score reduction for all injuries in EMS transports. Before getting further into the presentation, I want to acknowledge the role of myself, my mentor, and AI. For myself, I researched a lot of the past studies. I designed the study. I couldn't carry out the study because I don't have the licensing to do that, but then I worked on the data processing and analysis. My mentor oversaw the pilot trial and helped with the trials. He helped with the logistics of the trials and then helped to determine the data's meaning. I use AI to funnel research, finding articles that would help me with my studies, and then I use it for data processing. I worked with the East Pierce Fire and Rescue pilot trial, which was a pilot of 37 patients that were transported by the East Pierce Fire and Rescue Team. And all patients received a dose of IV Tylenol and where their pain scores were collected. And then the data was analyzed to identify the efficacy of IV Tylenol. For my exact methodology, it goes as follows. At the site, or the 0-minute point, the patient was identified, and if they permitted the use of IV Tylenol, they had to be conscious and respondent to do so. The infusion of IV Tylenol would begin with 1 gram in a 100 milliliter bag of normal saline over 15 minutes. Also, the first pain score was taken. At 5 minutes, the second pain score was taken, and the infusion continued at the 1 gram per 15-minute rate. At 15 minutes, the third pain score was taken, and the infusion was ended. At 25 minutes, we took a final pain score, and this was marked 10 minutes post-infusion. For my data processing methods, I categorized the complaints. I had 3 complaints that had 3 or more patients, so I decided to focus on these, which are abdominal pain, back pain, and headache. Pain score, I decided to average the pain score, grouping all the categories together to find the average pain score of each time interval, which was 0 minutes, 5 minutes, 15 minutes, and 25 minutes. Also, I made note of the 7-point threshold in which analgesia or pain relief is offered to patients with a pain score 7 or higher, and the goal is to get these patients under 7 points on the pain scale. For my results, I found the average pain score at 0, 5, 15, and 25 minutes. For the initial pain score, scores range from 7 to 10, as this was the threshold for analgesia, and the average pain score was 8.57. For 5 minutes, pain scores ranged from 5 to 10, and the average pain score was 7.46. At 15 minutes, pain scores ranged from 2 to 10, and the average pain score was 6.47. And at 25 minutes, pain scores ranged from 1 to 10, and the average pain score was 5.56. Also important to note, 31 of the 37 patients were under the 7-point threshold. Here I decided to isolate the initial pain score and the 25-minute pain score to see their differences. The average reduction between these 2 points was 3.01. And then my average pain scale reduction out of 10 versus complaint. These were the 3 main categories that had 3 or more patients that I focused on. Abdominal pain had an average pain score reduction of 2.25. Back pain had an average pain score reduction of 2. Then headache had an average pain score reduction of 4.5. Here are all the categories I used, even the ones that didn't have 3 or more patients. These were all included into the 37 patient study. The ones I did not already touch on were extremity pain, which had an average pain score reduction of 4.5. Facial trauma, which had an average pain score reduction of 2. Hip pain, which had an average pain score reduction of 4. Pelvic, which had an average pain score reduction of 3. And testicular pain, which had an average pain score reduction of 4.5. Discussing this data, I know that there's significant overall pain score reduction. All but one category reached under the 7-point threshold at 25 minutes. The one that didn't was back pain. Headache, which was another one of my main categories, had the largest reduction in pain score of those 3 main categories of 4.5. And with 4 patients, it gave me a P result of 0.2, marking it is significant. Then for abdominal pain, the average pain score reduction was 2.25. And this had 19 patients and a p-value of 0.001, which marks significant findings. With this study came a lot of limitations, as it was a very small trial size of only 37. This was due to difficulty with the pump, as it was a new equipment for the EMS team in the ambulance. So many people opted not to use this and seek other forms of treatment. And then there's patient refusal of IV Tylenol because it's not very well known. A lot of people opted for the normal opioids or no treatment at all. There's also very limited categorical data. As I touched on, there are only 3 categories with 3 or more patients in that category, so it kind of created a lot of scattering between different injuries. And then missing data points. There were variable transport times, so for the transports that got to the hospital earlier, it was hard to really follow up with those people when they're in the hospital, so I was missing 15 and sometimes 25-minute pain scores for them. And then adherence to pilot protocol. some of the EMS team forgot to ask for pain score times at certain intervals, which left blank in my data. Looking ahead for future research, I would want to create a larger trial with more patients, more data per category, more categories involved, and improve the ease of administration. This way, I could be more sure of my data and have more patients. I would create an in-depth study for headaches because 4.5 is a really large pain reduction. This could be comparable to opioids as an alternative. And then I would create trials to compare directly with opioid trials. This way I could get a side-by-side comparison and cut down on the speculation. Here are my sources and citations. And for my acknowledgements, I'd like to thank my mentor, Sean Wagner, who's the Assistant Chief of EMS at East Pierce Fire and Rescue, Mr. Garot, and the Marine Chemistry staff for helping me through these 4 years, the Marine Chemistry class of 2026 for being a great group to work with, and my family and friends. Thank you, guys. Do we have any questions? The question was, are there transports still providing patients with IV Tylenol? The answer is, to my knowledge, yes.
Your main concern was kind of offering an alternative to opioid treatment.
Yeah.
Did you have a chance to compare at all the pain levels with opioid versus the IV Tylenol?
Yeah, I didn't have a chance to compare them directly because it was really only one patient. I can't use both on them. But I knew it would be less of a pain reduction than opioids because they're a lot stronger. My thing was, would it get under the 7-point threshold to be a viable option if they were contraindicated?
You said you would like larger trial size. How do you think you would go about that?
The question was, how would I have gone about creating a larger trial size? I honestly think just doing it over a longer span of time. I only did 1 year. And also, like I said, with the pump and the other factors, getting more patients to be on board with this, and then having the EMS team be more comfortable with that, that would create a lot more patients.
When did IV Tylenol become like widely used or become like an option for
The question was, when did IV Tylenol become widely used for EMS transports? The answer is it still really isn't. This was really experimental. I think it was approved by the FDA in 2012. And yeah. Okay.
What was the difference in the pump that you're using in pump compared to what they have already?
The question was, what was the difference of the pump that was being used in the original pump? I don't really know, but from what my mentor said, a lot of the EMS team was struggling with the pump. I don't know what the differences were, but that was the thing.
What is the cost difference between opioids and IV Tylenol?
The question was, what are the cost difference between opioids and IV Tylenol? The cost is very similar. It's not really a factor. Up next, we have Dat Fan talking about bees.
Hi, my name is Dat, and I'm looking at the effect of queen caging and treatment timing on Varroa destructor infestation in honeybee colonies. So, why do honeybees matter? According to the USDA, honeybees, or Apis mellifera, pollinate about 75% of the fruits, nuts, and vegetables grown in the United States. In fact, 1 out of every 4 bites of food we eat is thanks to bee pollination. Altogether, that pollination adds to over $15 billion in crop value every single year. Because of this, honeybees are critical not just for food production, but also for biodiversity. However, bee populations are declining due to factors like habitat loss, pesticide exposure, and modern colony management challenges. That's why finding better ways to protect honeybee health has become an urgent issue. Research shows that one of the biggest threats are varroa mites. I chose this project because improving how we manage this parasite can help improve bee survival. So to understand why varroa mites are such a serious threat, it helps to understand the honeybee brood cycle. A worker bee develops from an egg into an adult over about 21 days. After the egg hatches, the larva is fed by nurse bees until the cell is capped. This capped brood stage is where varroa mites cause the most damage. Female mites enter the brood cell just before it is capped and reproduce in the sealed cell, shown in the image there. When the adult bee emerges, multiple mites merge with it, increasing the colony's infestation. Because mites are protected under the wax cap, many treatments are less effective during this stage. Because varroa mites depend on the brood cycle to reproduce, management strategies that disrupt brood production may improve treatment effectiveness. So that leads into the threat of varroa mites. Varroa mites, or Varroa destructor, are a tiny parasite that attach to honeybees and feed on them. While that alone weakens bees, the problem is further intensified when varroa mites spread viruses like deformed wing virus, which can seriously harm or kill entire colonies. Like I mentioned before, mites are protected by capped brood, shown in the bottom left of the picture, compared to uncapped brood, which is shown right above it. Research like Cornier and others in 2022 have shown high loads of mite lead to more different viruses, leading to a higher colony mortality rate. Because varroa mites reproduce under capped brood, not all treatments are equally effective. Oxalic acid is one of the most commonly used treatments because it is relatively safe for bees and effective at killing mites on adult bees. However, it cannot penetrate the wax cap of brood cells, meaning mites inside the brood are protected. Other treatments, such as Apiguard, can penetrate capped brood and target reproducing mites, having up to a 97% mite mortality rate. But they come with higher risks, including increased bee stress and mortality, according to Giacomelli and others in 2016. So if mites are protected by brood, then reducing brood during treatment may increase the effectiveness of safer miticides. Because many mites hide in developing brood, researchers have found that temporarily stopping the queen from laying eggs, called brood interruption, is a very effective strategy. One method is queen caging, where the queen is safely confined for a period of time, around 24 days. This allows the brood to emerge and creates a period where the colony is broodless. Studies like Gregork and others in 2017 have shown that during this time, treatments like oxalic acid, which can penetrate the brood, can reach more mites and increase the mite mortality rate compared to just treatment alone. So this leads into my problem. While previous research has focused mainly on the type of treatment or the season, almost no one has studied whether the time of day matters. So my objective is to determine whether applying oxalic acid after queen caging during the day versus at night affects the mite reduction. Since bees are outside during the day and inside at night, I hypothesized that night treatments after queen caging would kill more mites than daytime treatments. So it's important to clearly distinguish roles in this project. My mentor, Rick Salmon, led all technical beekeeping procedures. This included queen caging, colony management, oxalic acid vapor treatment, and mite sampling. My role in this was to assist him during fieldwork, develop research question hypothesis, and conduct the literature review. After data collection, I organized the data, performed the analysis, interpreted the results, and wrote all sections of the research paper. This ensured that technical procedures were professionally handled, and scientific design and analysis were student-driven. So to test my hypothesis, the study was conducted across 7 different apiaries in the Tacoma, Washington area over 2 separate summers. The map on the right shows the location, but the 2 of the apiaries were around the same area, which is why you can only see 6. Colonies were selected based on strength and brood size. So the general approach was to use queen caging, like the image shown on the right, for 24 days to create a broodless period. apply oxalic acid vapor, and then compare mite reduction when treatment was applied during the day versus at night. Varroa mite levels were monitored using sticky boards in a 24-hour time period to track. So to test the effects of brood interruption and treatment timing, colonies were divided into day and night groups. 2024 were day groups, 2025 were night treatments. All colonies received the same oxalic acid miticide treatment to isolate the effects of timing. Treatments were applied at 2 different times. Day were done 12 to 3 p.m. when colonies were at most fully active, and night treatments were conducted between 3 to 5 a.m. before sunrise. After treatment, mite levels were measured using sticky board placed inside the hives. These counts were taken days after treatments and represent post-treatment infestation levels, not the number of mites killed immediately by the treatment. So a lower mite count would mean a lower infestation level. So this graph compares varroa mite counts between colonies that received oxalic acid treatment only, represented by the no on the graph, and colonies that received oxalic acid treatment combined with queen caging, represented by the yes on the graph. This graph does not represent the hypothesis that I was testing, and is only to compare with past research on how queen caging affects mite counts. The y-axis represents the number of mites collected on sticky boards over a 24-hour period. Colonies that had undergone queen caging in addition to treatment showed a lower average mite count compared to colonies that received treatment alone. However, a t-test comparing these 2 groups produced a p-value of 0.13. Because this value is greater than 0.5, the difference was not that statistically significant. This indicates that within this dataset, adding queen caging to treatment did not result in a statistically significant reduction in varroa mite counts compared to treatment alone. This differs from past research where it shows that queen caging does have a statistically significant increase. But because there was extra variables, it couldn't be. This graph shows post-treatment varroa mite counts comparing colonies treated during the day versus night, which is what my hypothesis was testing. Each dot represents an individual colony, while the dot on the left shows the mean mite count, and the error bars showing plus or minus 2 standard errors of the mean. Colonies treated during the day had significantly lower post-treatment mite infestation levels than those treated at night. This difference was statistically significant with a p-value of 0.04. So what does this mean? I originally hypothesized that nighttime treatments would result in a lower post-treatment varroa mite infestation level based on the assumption that bees cluster more tightly at night, which could potentially increase exposure to that treatment. However, the data did not support this hypothesis. Instead, colonies treated during the day exhibited significantly lower post-treatment mite counts with a p-value of 0.04. A possible explanation for this outcome is that increased daytime activity may improve treatment distribution throughout the hive. During the day, bees are more mobile, younger bees are present inside the colony, and airflow within the hive may be greater, which all could better enhance the contact between the mites and the miticide. So why does this matter? These findings indicate that timing factors play an important role in treatment outcomes and should be considered alongside brood interruption stages. So there are several limitations that should be considered when interpreting these results. First was that mite counts were collected days after treatment. Measuring the data reflects remaining infestation levels rather than immediate mite kill rates. Additionally, colonies varied in strength, brood present, and initial mite loads, which may have contributed to variability within treatment groups. Another limitation of the study is that data were collected across 2 different separate summers. Although treatment protocols were consistent, the year-to-year difference in temperature, forage availability, and colony dynamics may have influenced mite reproduction. Also to mention, some colony used in the 2024 season did not survive into the 2025, which resulted in a difference in sample size across the seasons. So, where to go in the future? Future research should focus on isolating treatment timings effects within a single season to reduce environmental variability within years. Increasing the sample size would allow for a more detailed evaluation of queen caging as an independent factor, particularly when controlling for timing. Additional research comparing different miticides could determine whether the timing effect observed here applies broadly or is to a specific or certain treatments. And finally, examining colony behavior and airflow during treatment application could help explain why daytime treatments were more effective. So, in conclusion, the study addressed the gap in varroa management research by examining whether the timing of miticide application influences post-treatment mite infestation levels. Colonies were received miticide applications either during the day or night, and the results showed that daytime treatments combined with queen caging resulted in a significantly lower post-treatment mite infestation levels. These findings suggest that optimizing treatment timing may enhance varroa control without increasing chemical dosage or treatment frequency. So understanding when treatments are applied may therefore be just as important as how they are applied, which can offer beekeepers a practical and low-cost strategy to improve colony health. So I want to say thank you to Mr. DeGroat for being a great teacher and supporting and for guiding. My mentor, Rick Salmon, for being a huge help in the data collection, and I would have no clue what I would be doing without him. My family for constant support, Marine Chem class of 2026 for their feedback. Here are my references and any questions. Yes, that's my mentor, Rick.
This was an interesting experiment. It's clear that treating broodless hives is better and more effective. But it's difficult to find the sweet spot with getting the oxalic vapor crystals on the bees. And it seems to me that beekeepers have to be a little more flexible on their assumptions. Where the assumption was an evening treatment would be best because everybody's in the house. But it's what is the house? And we have no data on airflows in a honeybee colony that's cluttered with bees. So between day and night, it seems to me that there has to be this, like a sweet spot where treatment would be applied that would be most effective while we're keeping bees rootless. Just stating this is kind of an interesting outcome, and I will apply some of this.
Everybody give a round of applause for Rick for helping me out. The question was, did I tested my counts before the treatment? And there were my counts, but they were farther like before, so it would be a little inaccurate. So the mic counts were tested with white poster paper with Crisco applied onto it. It was stuck under, and mites that were on bees would fall onto it, and it would give a rough estimate for the infestation levels.
How did you come up with varroa mites as your primary study? Is that the biggest issue with honeybees?
Well, it was one thing that you can.
What's the question?
Oh, sorry about that. So he asked why varroa mites was the topic of my study for the biggest kind of factor, and I would just say that it was pretty easy to test compared to different things like pesticide exposure in like gardens, so and like climate change too. So. Any questions? Over here, somewhere?
Do you ever consider geographic variations or weather variations as a source of why there's variation?
So the question was, are there like geographic, weather, or environmental? And not that I didn't consider any of that. My apiaries were pretty spread out, which could be another limitation as well, but that was just not something I looked at.
Could you conduct this like in winter time or late fall or?
So the question was, could I conduct this in a different season? And yes, there have been like past studies where it was done during the winter instead of the summer. All right, next up is Sachin Suresh, investigating subtidal sea star populations at Titlow Marine Preserve, post-sea star wasting.
All right. Is there nothing up there yet? We're good now? Okay. Yep, that's me up there. And ever since I've been a little kid, I've been obsessed with sea stars. My name is Sachin Suresh, and for the past 2.5 years, I've been investigating subtidal sea star populations at Titlow Marine Preserve following sea star wasting disease. So what is sea star wasting disease? Well, in 2013, an epizootic, which is an epidemic among animals, spread up and down the west coast from as far as Baja, California, up into the southern tips of Alaska. And this impacted sea stars by first causing them to lose arms, then sexual organs, and then eventually die off, where they would take a kind of melting almost appearance shown in the picture on the screen. There have been several hypotheses about what the causative agent of sea star wasting disease is. However, in 2025, Prentice et al. Established that the current hypothesis is it is Vibrio pectinacida, specifically the strain which causes cholera in humans. So why study subtidal sea stars in the field? Well, this sea star project has been a historic project at the Bellarmine Marine Chem program, where we evaluate the health of the Titlow Marine Preserve against the sea star metrics. Keystone species often provide an outlook as to what the subtidal community or any community underwater and above water are looking like. So the studying of the keystone species that we looked at were the ochre star, the mottled star, the leather star, and the sunflower star. R.T. Payne's 1974 study established that the ochre star preys on mussels, as well as the leather star preying on urchins, which causes an impact on biodiversity. So without those species, biodiversity is decreased. The map on the screen shows the impact of the sites and all the sites on the west coast from Baja, California up into the southern tips of Alaska. So I just touched on the prevalent species, but in the top left-hand corner of the screen, that is the sunflower star. The top right-hand corner of the screen, you see the leather star. The bottom left-hand corner, you see the ochre star, and then the bottom right-hand corner, you can see the mottled star. So for my knowledge gap and leading objectives, previously the studies at Bellarmine conducted on sea stars didn't have a year-round data cycle, so we wanted the fall, winter, spring, and summer months. Previously, we just had the kind of late summer, early fall data set. So we wanted to assess the overall health of the Titlow Marine Preserve through the sea star metrics, and that was done through species count and then individual specimen size by species. So my investigative theories are as follows. I predicted the ochre and mottled star would increase, the leather star would remain stable, and the sunflower star would near extinction at Titlow Marine Preserve. The study area is depicted on the screen in the blue J-shaped transect. That is the historic Fairleigh pilings at Titlow Marine Preserve, and the green line indicates the swim-out path. The target species that I touched on, and the dives were conducted from September of 2024 to the September of 2025, all conducted at near slack tide, which is when there is no ebb and flow from the tide. It is either high tide or low tide. My materials and methods are as follows. I used an Insta360 X3 action camera to conduct underwater videography, and then once established that underwater photography provided a better method of data dissection, we switched to an iPhone 16 with a high-pressure adaptive underwater case. We used a meta ruler across 12 months, consistent, as well as standard scuba kit. I was in a wetsuit, and my mentor, Mr. DeGroat, was in a dry suit. The metrics we evaluated were, again, population count and individual specimen size. And then when I reviewed the photo and video documentation, the first review was for species count, and then the second review was for individual specimen size in centimeters. The student led all data analysis and measurements, and the mentor, Mr. DeGroat, conducted all of the underwater videography and photography, as well as accompanying me on my dives. Moving on to my results and discussion. So this graph right here is the 2024-2025 data cycle, visualized on the same y-axis, that is individual count. So that's 200 individuals is the top end of the y-axis. The leather star and ochre star in yellow and blue, those top two lines, show linear trends upward, showing recovery at Titlow Marine Preserve. The ochre star in red, closer to the bottom, fluctuates up and down a little bit, showing species sustenance at the marine preserve, but not necessarily recovery or decline. And then at the bottom of the screen, you can see the blood star and sunflower star. So I included the blood star in this graph while it wasn't a necessarily significant species to my study, just to indicate its presence at the marine preserve as a whole. So we saw it in the study area, but we didn't include it in the data sets that we looked at. And then the sunflower star is in green at the bottom of the screen, and that we saw one individual twice over the course of the study, showing functional extinction. Moving on, these four graphs show my data starting in the yellowish range to the blue range, aligned with previous and historic data sets from Bellarmine, ranging as far back as 2014. So in the top left-hand corner of the screen, the mottled star graph shows that linear increase with a huge spike to 177 individuals in March. The top right-hand corner of the screen shows sunflower stars with those two individuals we saw during the study in November and October, as well as previous studies having higher peaks at 14 individuals. The ochre star graph shows kind of sparse layout with jumping around, showing that there's not necessarily that recovery, but there's not necessarily a decrease. They're just sustaining themselves at the marine preserve. And then finally, the leather star on the bottom right-hand corner of the screen shows the steady increase yet again. One thing that's important to note about these graphs is on the y-axis, the scales are different. I couldn't overlay these graphs on the same axis because you wouldn't be able to see the sunflower star because the graphs are obviously a little bit smaller. I had to fit 4 on the screen, so that's important to note. As for my data analysis, the mottled stars showed the clear signs of recovery with a huge spike to 177 individuals, and that attacks seasonal recruitment. Now, seasonal recruitment is a term I'm going to start throwing around a little bit, so I just want to define it off the bat. That is all of the migration into the study area, as well as all of the birth rates in the study area. Leather stars also proceeded to increase beyond what was expected, with a strong survival showing, rather than short recruit pulses, a strong sustenance at the marine preserve. Ochre stars persisted in those low quantities, like I've touched on, not showing their recovery or decline, but rather sustenance. And then sunflower stars were deemed locally functionally extinct, which means that they're not fulfilling their ecological role at the marine preserve any longer. The loss of the top-down predation impacts biodiversity as a whole. So since the leather star and ochre star prey on both mussels and urchins and mussels kind of destroy kelp beds, if their populations become unchecked, that impacts all the way up to species like sea otters. So for my implications, the uneven recovery is reshaping subtidal communities. We're not necessarily seeing a return to pre-sea star wasting disease numbers, but rather a reset and establishing a new baseline data post-sea star wasting disease. The limited ochre and sunflower star populations have those impacts on the urchin and mussel beds, with the increasing mottled and leather stars displacing the other two stars at the marine preserve of Titlow specifically, as well as this has been shown across the west coast up and down. So I'm going to ask you to bear with me. I see a question mark on my next slide, which means that it did not upload properly. So I'm going to exit out and I'm going to get back in. Are we good? Okay. So the limitations that I want to talk about today is the biggest one right off the bat was our camera issues. So videography was a little bit iffy towards the beginning of the study. In October, we had to estimate the rest of the dives data because the back side of the J, the blue transect I showed earlier, we lost the SD card footage storage, so we weren't able to recover that footage, and we had to estimate based on mental count. So the October data was estimated. Additionally, I mentioned I was in a wetsuit and my mentor was in a dry suit. He would often laugh at me at the end of the dives because I would be freezing and very numb during December and January, but that resulted in us moving through the dive a little bit faster during those months just to make sure that I did not perish underwater.
So not really a limitation.
Not necessarily a limitation, no. The last thing I wanted to touch on is the single site focus. So several other studies from higher up organizations and universities have done cross-site comparison where they take the data and trends from several different sites and align them over the top of each other to see if the trends are consistent across the sites, or rather if it's individual based on each site. So for the applications and future research. Now this section I'm going to talk a little bit about the city of Tacoma. So in the past few years, the city of Tacoma has been removing the historic pilings around the coastline. And these are soaked with a chemical called creosote, and that's a tar and chemical compound to prevent the pilings from rotting away when they're submerged underwater. Now the site on the waterfront, the Mill Street Dock, I believe is what it's called, just had all of their pilings removed because these creosote-soaked pilings are proceeding to leach chemicals into the water around the pilings. So Titlow Marine Preserve is on the list of sites to remove the creosote pilings, and in this potential habitat loss for the sea stars is in the foreseeable future. So my study set up a baseline data of a full year cycle so that a future student could come through and using the same kind of coordinates that I used, go back and view if the habitat destruction that is caused by the removal of the pilings is statistically significant enough, or the creosote leaching is statistically significant enough to remove the habitat. The last thing I want to touch on is the University of Washington's Friday Harbor lab programs. So they are breeding sunflower stars, the functionally extinct species that I've touched on, actually in their Friday Harbor labs, and then releasing them into the wild. So just recently they released 29 individuals into Friday Harbor to regulate urchin beds. And so there is a glimpse of hope for sunflower stars making a return on the West Coast. Here are my references. And I'd like to extend a huge thanks to Mr. DeGroat for mentoring me. I've been kind of a pain, I know that. Huge thanks to Ms. Eder and Ms. Nuno for nitpicking me on my slideshow and making sure it looked pretty. My mom, dad, and sister, of course, for listening to my endless presentations late at night. My friends for providing the support when I needed it most. And of course, the Marine Chem class of 2026. Thank you for all the memories. All right, any questions? Yeah.
Why were the sunflower stars most affected?
So the question was, why were the sunflowers most affected? Some of the stars, so the leather star in specific has a mucousy kind of coating on the outside, and that reduces transmission rates. And then the ochre star and mottled star were both seen as not really having too much of an impact over the course of the years since 2013. They kind of just never dropped off the same way. I believe it's just bacteria susceptibility in sunflower stars, as far as I know. Yeah.
How did you identify that they were declining due to sea star wasting disease and not another factor?
So the question was, how did I determine that the sea stars were declining due to sea star wasting disease and not another factor? Sea star wasting disease has been the major kind of plague among sea stars as a whole up and down the West Coast. It's been exclusively the cause of mass die-off. Previously, sunflower stars were the most prevalent species at Titlow Marine Preserve. When I was a little kid, you saw that picture of me holding the mottled star, but when I was a little kid, sunflower stars would kind of coat the entirety of Titlow Beach during low tide. So seeing them in this low of quantities kind of establishes that this is due to sea star wasting disease and not necessarily due to another factor.
Can you show the sea star wasting picture one more time?
I can. Let's see.
Part of what you see.
So like I touched on, they take on kind of a melting look to them as they kind of like almost disintegrate. So that purple star right there is kind of taking on that melting disintegration look. Any other questions? Yeah.
Do you notice any differences in populations and depths when you have to go deeper?
So the question was, did I notice any differences between population at depths? So our study remained kind of in a pretty relatively similar depth range. Had I done a cross-site comparison, I may have seen differences in depths, but because of the way that our study was set up, I didn't see any. Yeah?
Did you identify any sea stars that were actually affected by wasting disease in your study?
So the only one that I remember that we kind of notably underwater made a big deal about was the sunflower star when we saw it. One, because it was so rare, we saw it twice, the same individual. And so the first time we saw it through, it had some color to it. The second month it was colorless and gray, and it seemed to not be moving as much. It was in the exact same spot actually. So that was kind of our signs, and then the month after that we didn't see it. Any other questions? Yeah, right here.
How long does it take to grow a sea star?
How long does it take to grow a sea star was the question. So I actually have no idea. The Friday Harbor labs have a pretty tight lockdown on their lab growth processes, so we don't have access to any of that. I have no idea how long it takes to grow a sea star. What's up? I see a question in the back.
Yeah, within the data collection, like, do you have a process for ensuring that you saw all the specimens that like were in the area, and do you have like a certain like margin of error for how much that would come off as month to month, or like what percentage do you think you got of within this area?
So the question was that how was I sure that I saw all of the sea stars in the area, and then how did I calculate percent error? So over the course of the years, the study site has, the study zonage has increased from about 6 by 6 feet underwater to now 12 by 8 foot is what we did this year. And so we didn't necessarily, when we would go through some months, the current would be picking up a little bit and the seaweed would be blown over down on the ground. So there really was no way for us to ensure that we got every single sea star. Again, other months I was freezing, my feet and hands were about to fall off. So we moved through pretty quickly, and then I did not calculate percent error on how many I missed, because we didn't have an estimate of like the total number of sea stars in the given area per se. One more question? Last question, yeah, go ahead.
Do you predict the sea star population between species? Will the data will go back to how it was before sea star wasting as time goes on, or do they go to fluctuate and rebuild entirely?
So the question was, do I predict the data moving forward in the future years will align more closely with the data before sea star wasting disease, the disease was introduced? So I do think that with enough time, certain species will align closely with the trends that they had previously. Sadly, I can't say with my whole heart that I think sunflower star is going to make a recovery. I don't believe that they will. Albeit with the lab efforts of Friday Harbor and UW, I don't think that they're going to have the kind of results they're looking for. And then as for the other populations, I think leather and mottled stars are going to continue to increase, and we'll probably see ochre stars get pushed out a little bit just because of resource competition. As those populations kind of expand beyond bounds, ochre stars are going to get pushed out is what I believe is going to happen in the future. All right. I'm not going to introduce another person right now. We're taking a 10-minute intermission. So thank you so much.
Hey guys, so this is don't forget to get your wristband.
why don't you go ahead and get on. I'll introduce you next time. So if you see some folks outside, why don't you yell at them, coax them back in, please. All right, so to finish this off, we have three more presentations, starting with Marie, who's going to talk to us about bees again, but in a little different light.
Do we actually need to save the bees? You've heard and seen Save the Bees thousands of times over the past decade, and earlier tonight. Throughout the country, we have seen a decrease in bee populations. But is the problem truly that we don't have enough bees, or that we're not utilizing them in the correct way? Although factors such as overuse of pesticides, habitat loss due to urbanization, and parasites should cause concern for citizens, is there a way we can better use the bees we have now? Hello, my name is Marie Wigman, and my project is training bees to change their pollination habits through electromagnetic fields. As we look at this graph from Illinois' Farm Doc, the orange bars represent the total honeybee colony supply for all of the United States. And in blue, the number of colonies needed for the almond groves in California. Every year, the gap between the blue and orange has shrunken more and more, despite increased numbers of colonies, as seen in the steady rise of the orange bars. The rest of the United States doesn't reap the reward, as most of these colonies are transferred to California. So where are these bees coming from? There's not an adequate amount of food sources in California to sustain these 2 million bees, which means they're being transported into the state through commercial beekeeping operations. Although pollination begins in February, the preparation starts months in advance, even as early as the summer before. This includes making sure that colonies are strong enough through treating for pests and diseases such as varroa mites and supplementing diets. These high costs lead to farmers needing to put in $200 per colony. This method is not sustainable for the rest of the farmers in America. Hence, the great migration of bees to the almond groves in California. This tension is exactly why new ways to shape bee pollination is so necessary. The farmers of America should have enough bee colonies to effectively pollinate their crops, but they aren't able to utilize the bees that they do have in the most beneficial way. The same situation applies to California farmers. They don't need to fly in as many hives as they do. They need to find a way to use the native bees of California to their advantage. And this problem doesn't just affect the rest of the country. This problem affects our own community as well. The Bellarmine greenhouse is a thriving aspect of our campus life, but along with most of the country, is also struggling with pollination. The issue the greenhouse is facing is very similar to what is happening to large-scale farmers. They can't seem to get the native bees of the area to pollinate the plants they want or the spaces they want pollinated. For the greenhouse, this means that the beehives on the north side of campus aren't motivated to pollinate within the greenhouse. Many plants are kept within the greenhouse in order to keep a stable environment. The downside of this is that the bees don't enter the space. This means that the plants are currently being hand-pollinated. If we can find a way to shape bee pollination, we could encourage the bees to enter the greenhouse. The newest research showed that the answer to this problem may lie within technology. Enter EMFs. Electromagnetic fields, more commonly known as EMFs, surround us every day. They are simply put a combination of both magnetic fields and electric fields. Both the sun and the earth are considered EMFs, but more common items such as power lines, phones, and Wi-Fi are also EMFs. The ability for an organism to be able to pick up on EMFs is known as electroreception, which has been known to found in aquatic organisms for years. It has been studied more closely in recent years with bees. Gregors et al. 2013 was able to find that honeybees utilize the joints in their antennas to sense EMFs, while Clark et al. In 2013 found that other types of bees, like bumblebees, may use small hairs to detect the fields. Roberts et al. Was able to use a reward-based system to train bees using electromagnetic fields. Differences in strength and electric potential change between plant species, but all rely on EMFs for pollination. It has been shown that bees can be trained to learn that when EMF is present, they will get a reward, and that when one is not present, they get a deterrent. Roberts and Clark were able to do this through setting up 2 posts. One with a positive charge would have a reward, like honey, waiting on top for the bee to pick up. The one at ground level would have a deterrent, like vinegar. Over time, the bees learned that if they went to the post with a positive charge, they would be positively rewarded. After the training period, the rewards and deterrents were removed, and just the posts were left. The bees continued to be attracted to the positively charged post and avoided the ground level post. Mrs. Savage, the lovely woman who runs our greenhouse, saw this research and wondered if EMF training could be used to benefit the Bellarmine campus. The problem I was trying to answer was, can bee pollination habits be trained in order to change how bees naturally pollinate the plants in their habitat? The objective of my research was to train bee pollination using electromagnetic fields. And my hypothesis was that when bees are trained to notice patterns in electric fields, their pollination habits can be shaped to direct their pollination to specified areas. To provide you guys some context of the space in which we conducted this research, this is a photo of the northwest side of our campus. I'd like to direct your attention to the yellow arrow. This indicates the space in which we trained the bees. The red arrow indicates the space in which the testing of the training was carried out. These locations are known as the training phase and the testing phase, respectively. To train the bees, close to the hive, 2 identical 5-foot aluminum posts with a 1.5-inch diameter were set up in planting pots with the same levels of river rocks placed in each. Placed on top of each post were 2 matching epoxy plates. One post was charged to 25 to 35 volts using a Ward's 365112, while the other was kept grounded. The voltage was chosen based on the previous study carried out by Clark and Roberts. 30 volts allowed for the post to be charged enough for the bees to sense the field without increasing the risk of harm to the bees. The post that was charged had a sample of honey placed on top in order to reward the bees when they visited the charged post. The grounded post had apple cider vinegar placed on it in order to deter the bees once they visited. This process continued for 5 days before the next step of the research began. The major difference in our method was the use of rocks and plastic chicken fencing. The rocks provided high points for the bees to land on to ensure they did not get stuck in the honey to ensure full safety of the bees. The chicken wire, although originally intended to deter fellow students, ended up creating a concentrated EMF, as we later found out that the chicken wire was an EMF blocker. This means that the resin disc at the top of the post was where the EMF ended up being circulated to. Our power source had a magnetic field strength of 3.4 microteslas, while our epoxy disc had a reading of 0.5 microteslas. The middle part of the post had no reading. This means we essentially made a Faraday cage. The purpose of a Faraday cage is to protect an area from electromagnetic radiation, which for us meant that the part of the post that the plastic chicken wire covered was not emitting an electromagnetic field. The level of microteslas emitted assured human safety along with bee safety, with concerning levels being over 100 microteslas. Just bear with me for a second. I'm going to try and reload this. Maybe? So sorry, everybody. Okay, there we go. After the bees had been trained for 5 days, the post that was electrically charged was moved next to a popular pollinating plant. The reward system was taken away, leaving just the EMF to attract pollination. Prior to the training period, photos were taken daily of the pollinating plants in order to determine a baseline of pollination for the plant. In a similar way, every day at approximately the same time, a photo is taken from the same angle in order to record the number of bees present at the given moment. We chose to keep the chicken wire in order to continue to concentrate the EMF. On the left, which you guys unfortunately cannot see, we have the data from our control group. During the first 5 days, there was not visible bee pollination for the plant, leading to the 5 dots that were shown on the left. After the EMFs were introduced, this number shot up with pollinators seen on every day, with 4 days having 2 bees and the second day having 1. Although these numbers may seem low, post-introduction of the EMF on a typical smaller pollinating plant like the one we used, there's typically seen 1 to 3 bees per day of pollination. Given that each data collection captured half of the Joe Pye weed, these numbers align with typical pollination habits of our bees. I utilized GraphPad, which allowed me to plug in my results, and it gave me important values for a 2-tailed t-test, which I found was the best test to run for my data. After performing this t-test, the results showed that our p-value was 0.0001. This p-value is below 0.5, which means that our results are statistically significant. Our limitations included that we started later in the pollination season than we would have hoped. Peak pollination time is earlier in the summer, but we chose a plant that pollinates later in the summer to try and combat this. This is also a pilot study on the Bellarmine campus. This means that there was a small sample size and limited resources. The centralized EMF cage was not a part of our original plan, and we didn't realize how concentrated the EMF was until after the study was completed. Due to this, we also don't know the location of the charge that the bees were attracted to. We know that both the top and the bottom of the post was charged, but we don't know which part of the system created the positive charge that the bees associated with the reward. Since we did not mark each bee individually, there is no way of knowing if the bees we trained are the same bees that ended up pollinating the Joe Pye weed during our testing. And finally, we have 2 different species of bees within our 3 hives on the campus. We have both Apis mellifera and Apis carnica. There could have been different effects of training on the 2 species, with one responding well and one not responding. In a similar way to the last limitation, we do not have a good way of knowing which species was pollinating the Joe Pye weed. The results do support my hypothesis that EMFs can be used to change bee pollination habits. Having a p-value below 0.05 shows that there is correlation between the training and the pollination of the Joe Pye weed. Due to the small sample size of the research, there's still a high probability that the results could be left up to chance. My research supports past research in this field. We were able to replicate the study of Roberts and Clark and build onto it. We took their training model and applied it to real-world pollination. These findings can be used on the Bellarmine campus, but could also be applied in large-scale farming. Future research could work on moving the pollination indoors. We utilize the plant outdoors as a stepping stone toward moving indoors. But the eventual hope is that all parts of our garden can be properly pollinated. In addition to this, a rotating training system that has a training station set up on the far north side of campus with the original reward system and the lone positive charged EMF station set up could create a long-term solution in addition to creating more data to analyze. I'd like to thank Mrs. Savage and the Bellarmine Greenhouse, Anna Shimshock, Claire Walsh, and Lila Holst for going above and beyond to help me with my data collection, Mr. DeGroat and the Marine Chem program, my parents and family, and of course, the Marine Chem class of 2026. These are the references I used for this presentation. Thank you very much. Do I have any questions? Yeah.
How long does a pollinating bee live?
The question is, how long does a pollinating bee live? I am actually unsure of this. We were able to conduct our study within a 2-week range. So our hope was that most of these bees were the same ones that we originally tested and trained, but I'm not quite sure of the exact lifespan of a typical pollinating bee. Oh, yes.
How can you tell that the training actually did something versus the bees are just innately attracted to the EMF? Zero control?
Yes. So the question was, is there any way to tell if the training is what attracted the bees to the plant or if this is just a typical level of pollination for this plant? We were able to create a control group a couple weeks prior to the testing, which showed that there was no pollination for this plant for the 5 weeks which we took data, for the 5 days that we took data. And following the training of the bees, the following 5 days of data collection showed that every day had pollination of the plant, which is in line with a more typical amount of pollination for plants in their peak pollinating season, which since we were at the later end of pollinating season showed that there was a correlation between our training and the increased amount of pollination to the Joe Pye weed. Yes?
What is the difference in comparison between the original experiment that was based on the Robert one or whatever and your results?
Yeah, so the question was, what are the differences between the original study and my study, right? Yeah. So Roberts and Clark were mainly focused on seeing if bees could be trained to sense the difference between an electromagnetic field and something that's held at ground level or has no electric charge to it. And so they didn't apply their training to see if they could actually increase pollination to a plant. So we utilized their original study to create the basis for our training system. And so we modeled their original idea of having a post at ground level and a post that was charged and creating a ward-based system. And then we took that positively charged post and were able to put it next to a plant to increase that plant's pollination. Okay. Oh, yes. The question is, how costly would this be? The nice thing is, if this is on the Bellarmine campus, a lot of the tools we had were already available at Bellarmine. So the battery that we used is part of the physics program at Bellarmine already, and now we have the supplies to do everything else. So it actually cost very little money for us to do it. On a different location, it might cost more money. The robotics program was also kind enough to donate a lot of supplies to use to create the training system. But it cost around, I think, $50 for the original, for the new material we needed that wasn't already supplied at the Bellarmine campus. Yes. The question was, what would I have done differently to work around the limitations? My main thing was I would have started earlier in the pollination season. If I had started closer to May and earlier in the spring and summer season, we probably would have had more data to collect on the levels of pollination. And I would have also created a longer training system with more steps. So the original goal was to move the pollination system inside the greenhouse, but due to it butting up with the end of the pollination season, we weren't able to do so. But I highly recommend for future research. Thank you very much. Next, we have the beautiful and talented Claire Walsh to discuss the impact of closed environmental agriculture, CEA, on the quality and taste profile of tomatoes.
Well, for many of us, access to food is as simple as going down the street to your local Fred Meyer or maybe Trader Joe's. In reality, food insecurity is a major issue across the world. And while it's a very multifaceted issue, one of the main causations of food insecurity is demonstrated in the map you see above you. Quality farmland is not an unlimited resource, and in many parts of the world, like northern Canada or sub-Saharan Africa, there can be entire land masses or even countries that have very little quality farmland at all. This can make access to fresh food limited and resource intensive. However, recent innovations in the agricultural industry have arisen as a possible solution to this problem. One of these such innovations is controlled environmental agriculture, or CEA, as you might hear me refer to it. CEA generally refers to greenhouses and plant factories, such as the Bellarmine greenhouse, for example, often utilizing non-soil systems like hydroponics, aquaponics, or aeroponics to grow plants. Most of the time in these systems, plants receive light from artificial sources as opposed to traditional sunlight, and this will be the primary focus of my study today. But how does this shift towards closed environmental agriculture affect food quality and overall consumer experience? Hi, my name is Claire Walsh, and I'll be talking about investigating the impact of closed environmental agriculture on produce quality and taste profile of Solanum locopersicum, which is commonly referred to as tomatoes. I based my research around 3 main research papers that were previously published. Baldwin, who discovered that fruit quality is defined by biochemical traits such as sugars and acids, which I based the majority of my data collection off of. Smith, Lee, and Chang determined that light spectrum and daily light integral greatly influence flavor and nutrition. Light spectrum is things like color of light, such as red light versus green light, and daily light integral is the amount of hours of sunlight per day a plant receives. Fernandez discovered that consumer preferences greatly influence scientific research and agricultural practices, pretty much saying people care where their food comes from. So the problems and objectives I hoped to address. My main problem was that there was very little research comparing how different aspects of closed environmental agriculture, such as lighting or hydroponics, directly affected tomato quality. Secondly, I wanted to bridge the gap of how two different varieties of tomatoes may react differently to these variables. My main objective was to evaluate how differences in light environments affect tomato flavor. I had two main hypotheses. My first one was that there would be a significant difference in fruit flavor rating when comparing natural and artificial lighting. My second hypothesis was that differences in flavor rating will be less pronounced in hydroponic systems and cold weather varieties than in soil-based systems and warm weather varieties. So I followed the main general procedure for growing tomatoes, which is somewhat of a laborious process, but pretty much you germinate the seeds in either soil, or I used a very specific rock wool germination for my hydroponic plants. And then they were transplanted into their test environments, which is either their pots if they were one of the soil plants, or the hydroponic unit if they were the hydroponic plants. Third, they were fertilized and pollinated. Our soil plants were pollinated with a solid fertilizer, courtesy of our Bellarmine chickens, and our hydroponic plants were fertilized using a liquid fertilizer that was bought by the Bellarmine greenhouse. Each of these were hand pollinated, as Marie kind of touched on in her presentation, but because a lot of these plants were inside, I had to hand pollinate them. Our fourth step was harvesting and preserving for data collection. Pretty much, I just had to determine when a tomato was at its peak ripeness, and then I would pick it and then use it and process it for chemical analysis. My chemical analysis mainly consisted of pureeing the tomato sample and then running it through a pH electrode, which gave me a pH value, and then I would filter the puree itself into a liquid, which I could put into the refractometer to get a Brix percentage, which is a measurement of sugar. So I ran two main experiments. Experiment 1 started in about May, I would say, and experiment 2 started in August. Experiment 1 was mainly focused around a warm weather variety tomato called the Delicious variety tomato, and were my soil plants. So I had two main groups, which is our natural soil group, or NLS group, and our artificial light soil group, or ALS group. Again, I used solid fertilizer for this group, and the main thing I was comparing between these two was the natural and artificial light. For experiment 2, I used a cold weather variety plant. Again, I used rock wool germination, which is a very specific germination technique used for hydroponic plants. And I was again comparing our natural light to our artificial light with our NLH plants and our ALH plants. Just to clarify, these experiments are not directly comparable because they have different varieties and different growth environments. So mainly I'm just focusing on how the natural light and artificial light, the plants react differently to those two factors. So for my flavor rating, which is pretty much how I ranked these tomatoes, I evaluated each tomato based on two key qualities. Their acidity, which I recorded as a pH value, with the lower the pH value, the higher the acidity, and their sugar content, which I recorded as a Brix percentage, which is a measurement of the sugar percentage in the liquid of the fruit. Using these two qualities, I created a ratio labeled as a flavor rating, following the principles demonstrated in this infographic. A high acidity, high sugar content tomato is generally a good tomato. By dividing Brix percentage by pH, I created this flavor rating that demonstrates both acidity and sugar. The higher this value or flavor rating, the better, pretty much. So for key results, across both experiments, acidity remained very constant, but sugar content varied very meaningfully, especially in my natural light plants, which is why you see such a large range for my purple plants, which the purple dots, which are the natural light plants, as opposed to the bluish green dots, which are our artificial light plants. The top graph is our first experiment, which had considerably less tomatoes than my second experiment. We had about 3 tomatoes for each of those plants for the first experiment, and then my second experiment, which is the bottom graph, we had about 9 or 10 tomatoes for each plant. Experimental value experiment 1 had a p-value of 0.26, which means there wasn't a statistically significantly different flavor rating for our natural versus our artificial light plants. And experiment 2 had a p-value of 0.1, which demonstrates some correlation, but again, not inadvertently statistically significant. So overall, neither of these were statistically significantly different between our natural light and our artificial light, but there is definitely some correlation you can see. And the main reason for that is because there was such a big variety of flavor ratings between our natural light plants, which kind of makes sense because natural lighting is variable, whereas the artificial lighting, they were receiving exactly 12 hours of lighting under a lamp every single day. So across both experiments, I combined them in this other graph. Even though they're not directly comparable, it's nice to see them put up right next to each other just for comparison. There was some statistically significant difference between the two, but it more actually indicated that there wasn't a difference caused by the lighting, such as natural versus artificial lighting, but instead the statistical significance was from the environments and varieties themselves, which I think is really interesting. Pretty much my data was concluded that the environment had a much more key factor, and the variety of tomatoes, such as cold weather variety versus warm weather variety, than the lighting. So to discuss, neither of my hypotheses was directly supported, but hypothesis 2 was partially supported because of the differences in the hydroponic experiment was less pronounced than the soil experiment. But the greatest find from this was that other variables like our growth environment or our variety had much more of an impact on the flavor rating than lighting, which was unexpected. So some future implications, obviously we can take this knowledge that we have now and put it more into how we grow food in our Bellarmine greenhouse, specifically utilizing our hydroponic units more. This adds to our sustainability that we are very adamant about at Bellarmine for growing food sustainably, and since we don't have a lot of viable soil on campus, the hydroponic is a great way to combat that. And also just broader implications for the agriculture industry. As I said before, farmland is not an infinite resource, and being able to know that controlled environmental agriculture could be a possible solution and doesn't have a huge effect on produce quality is very important. And then additionally, just consumers being able to know that the food quality isn't tainted because of how it's grown, if it's grown in one of these controlled environmental agriculture places. So it's kind of just peace of mind with that. I had a lot of limitations with the study. Specifically, I had to restart experiment 2 over because I got wrong seeds, like they weren't the seeds that they told me that they were, so I didn't get a single tomato out of my first batch, so that was a little frustrating. Also, just in general, growing plants is not very easy. I had a lot of issues, and I'm sure Ms. Savage can attest to that. But it's also just a lot of work. I had to go in almost every other day this summer just to water them and stuff like that. I also had issues with rodents eating my tomatoes before I could harvest them, which was really frustrating. And partially, that was the reason I only got 3 tomatoes each plant from my first experiment. And also I had the power short circuit in my hydroponic unit, which was kind of a big deal because the pump stopped working, so I had to go in and manually fill up the water almost every single day. And also Ms. Savage helped a lot with that because the pump would not pump water to each hydroponic level. And also for the chemical analysis, I originally wanted to send my tomatoes to get tested by an actual food science center. And me and Mr. DeGroat reached out to multiple places, and none of them would test my tomatoes for me, at least not without costing thousands of dollars. So I ended up having to do that myself, and I didn't really have a lot of the equipment. And I wasn't really able to test for a lot of the things I wanted to test for, such as vitamins, and I wanted to run an HBLC test, which I wasn't able to do, which would have been further insight into the exact quality of our tomatoes. So for future research, I think there's a lot of potential future marine chem projects, specifically, I think variety-specific tomato projects, such as cold weather versus warm weather varieties, and looking at that individually. Also comparing hydroponic, aquaponic, and aeroponic systems would be really interesting, and also just using other plants in a study similar to this. So some acknowledgements. I want to shout out to Miss Savage. She helped a lot with growing my tomatoes. It was a lot of trial and error. And also Mr. DeGroat for giving me a lot of insight and helping me find most of my sources. Bellarmine facility and staff for being somewhat okay with me being here all the time during the summer. Key references and authors. The papers I used were really impactful and shaped my research a lot. And also just my friends and family for supporting me. And of course the Marine Chem class of 2026. These are my references. Are there any questions?
Did the different varieties of tomatoes affect the sugar or pH levels or something?
Yeah, so the question was were the different varieties of tomatoes affecting the flavor rating or the acidity-sugar ratio? So because there was some difference between the two experiments, but it wasn't because of the lighting, it can be inferred that it was because of either the variety or the way they were grown, so like soil versus hydroponic, but we can't really tell if that difference is because of the variety or because of the growth environment. So that's definitely something I would have wanted to test for, but we just can't really tell with the multiple variables that were in play. So the question was, if I was to do a follow-up experiment, what factors would I focus on? I think that was what it was. So because, like I said, the lighting didn't really have an impact on our flavor rating, but other factors might have. I think looking specifically at those factors and seeing which one it was, I think would be really interesting, whether it was the variety or the growth environment. If I were to repeat this experiment or do it again, I would focus on one of those to see if that's what made the difference between the two experiments.
Is there any specific environmental factors that you want to focus on or test to see if they have a direct impact?
The question was if there were any specific environmental factors that I wanted to test for. I think definitely seasonal factors are really important. Tomatoes are very seasonal plants, and especially because we had the opportunity of using the greenhouse, there is the opportunity to grow tomatoes year-round, which would be really interesting to see how the seasons affect it. I think that would be very interesting. And also environmental factors such as pollination, it would be really helpful if they were naturally pollinated and to see how that affects it versus me having to hand pollinate all the plants. So that could definitely play a part in the flavor rating itself, yeah. Could you repeat that?
How did you determine if a tomato had been ripe?
So the question was, how would I determine if a tomato was ripe enough to be harvested? I think that's what it was. I watched a YouTube video on how to see if it was ready, but mainly it just had to be all red, and there's like a certain texture of a ripe tomato that you look for. But I had to be really careful with leaving my tomatoes to ripen too much because the rodents would come and get it.
When determining like your flavor rating, did you get a taste any of your tomatoes, or was your final results only based off of like the objective data?
So the question was, did I get a taste any of my tomatoes, or was it only based off of the data itself? So originally I wanted to do a consumer panel, so also have my data, but also maybe like have fellow classmates try them and rank them. But unfortunately I just didn't get enough tomatoes to do that, and also I really don't like tomatoes.
So you got no information, feedback from the rats?
The rats did not want to be surveyed, unfortunately. Up next is the beautiful, amazing Genevieve to talk to you about coral reefs.
Genevieve's going to sit there.
It's going.
Oh, you really cannot see anyone come up here. This is really fun. Okay, hi everyone. Good evening. Thank you for being here. I know you're probably a little bit overwhelmed by the amount of information that is being presented to you tonight. So we're going to start with something a little bit interactive. I want everyone to hold up their pinky for me. And then just take a look at it for a second. A flatworm of that size can kill an entire coral colony in a matter of weeks, which many of which are decades or thousands of years old. I'm Genevieve Davis, and my research was aimed at comparing pest-killing treatments in a native Hawaiian coral nursery. Coral reefs make up less than 1% of the entire ocean floor, and yet they are the most biodiverse ecosystem on Earth. Mainly found in tropical and subtropical waters in the Indo-Pacific region, reefs provide protection from storm surges and erosion, and play vital roles in supporting human life through fishing, recreation, and even medical research. Despite this, these ecosystems are becoming increasingly fragile and are estimated to continue to face accelerated global decline in the next following years. According to a study done by Baker et al. In 2018, with the increase of oceanic warming and acidification, an enormous amount of stress is placed on coral skeletons. Oh my gosh, coral skeletons. This kills off photosynthetic algae, which serves as coral's food source and leads to bleaching and discoloration. As a result, reefs may become more susceptible to parasites and disease, which feed off the remaining healthy coral and eventually leading to death. Data collected from the State of Ocean report in 2024 show that in the past 20 years, there have been 4 major mass bleaching events. These main events are caused by water increases by just to 1 or 2 degrees Celsius, and each time they have left more dead coral behind. Today, about 54% of healthy coral is remaining globally. So to combat this, coral nurseries around the globe, which are essentially seen as underwater farms, are working to retrieve corals from damaged reefs and bring them back to facilities to ensure growth in a stable environment. Once corals return to a healthy state, they are replanted and continually monitored to ensure reef regeneration. Specifically, the nursery I worked with was the Hawaii Coral Restoration Nursery on Sand Island, Oahu. Hawaii is one of the highest rates of endemism, which is when species are so native to one location that they can be found nowhere else. And while this applies to many mammal, fish, and bird species on the island, it also extends to corals. Due to this, the fact that the native Hawaiian corals are one of the slowest growing reef-building corals in the world, the nursery is highly involved in the protection and conservation of the current reef populations that protect and provide ecosystems with life. When corals come into any nursery, they are treated with certain dips to remove any outside pests that may be on or in the skeleton to prevent further inhabitation of the corals that are currently being in quarantine. At the Hawaii nursery, 2 dips are currently being used. The first is Lugol's iodine, which is an iodine-based treatment, and the second is Revive, a treatment that consists of plant extracts and is good for newly cut corals. In the past decade, an additional treatment, hydrogen peroxide, has been introduced to the coral community, but is not being currently used at the Hawaii nursery. In a study done by Shawkat et al. In 2011, hydrogen peroxide has been shown positive results, but has also led to coral bleaching and tissue damage. The study also showed that soft tissue corals are more susceptible to damages that may result from using peroxide as a dip. While all treatments have been proven effective to some capacity, there are still gaps in knowledge in the effectiveness of said dips being used at higher concentrations or combined with other treatments, which led me to asking 3 questions. What would happen to the coral tissue if I were to double the concentrations of the currently used dips of iodine and Lugol's? What would happen to the corals if I combined the commonly used Lugol's and Revive dips to create a new mixture? And how much damage would occur if I were to introduce a hydrogen peroxide treatment to the corals at both base and doubled concentrations? So I first hypothesized that doubling each iodine and revive dips would not cause any significant damage to the coral tissue. Secondly, that the Lugol's and revive mixture would result in the least amount of damage out of all the treatments that were being tested. And finally, that the doubled concentrations of hydrogen peroxide would cause the most damage to all species. For my research, I spent a week in Hawaii and worked with my mentors at the coral nursery, Christina and Nicolo. They collected the coral samples a month before I had arrived at the nursery, and I helped with cutting each sample into fragments and creating each treatment. I performed 24-hour pictures and observations, and a week after I left, my mentors collected follow-up pictures and base-level observations and sent them to me. For the setup of my project, it was broken into 3 main parts. First, my mentor and I took corals from quarantine tanks in the outside portion of the facility, cut each into fragments, and transported those fragments into tanks within a UV-lit room where the experimentation took place. The 3 species we sampled from were the MCA, PEV, and PLO species, or commonly known as the great star coral, the cauliflower coral, and the lobe coral. For each species, we took 2 samples, which were each from 2 separate dive sites. From each sample, we cut 16 1 by 1 centimeter fragments so that 2 could be used for each treatment. So this is a list of the 8 treatments that we performed experiments with the first 3 being known and frequently used at the nursery, and the rest being our experimentation treatments. Finally, pictured there on the left is a visual to show the coral colonies we sampled from and their fragments next to them. And then the picture on the right shows the tank setup for 1 treatment. So you can imagine this setup basically done 8 times, and they were all lined up next to each other in a blue-violet UV lit room to stimulate growth. So to apply our results to both the short and long term, we used data 24 hours and 1 week post-treatment. For each of these time frames, I conducted 2 forms of analysis. The first was a simple visual inspection of the corals, and I ranked them on my own scale of 1 to 5, with 1 meaning said treatment caused little to no visual damage, and 5 meant very severe or death to the coral sample. The second was an image processing software called ImageJ, which measured the area of discoloration on a coral fragment and compared it to the total area to determine percentage of mortality. So when looking at graphs, we first have the great star coral. The y-axis shows us the severity ranking, while the x-axis displays the coral species. And each dot on the graph corresponds with the treatment being used, which can be seen in the key in the top right-hand corner. At 24 hours post-treatment, the only dips that showed little to no visual damage were the seawater control, the base iodine dip, and the base revive dip. But now looking at this cluster of treatments, we can immediately see that the remaining dips that were being tested rank all above a 3, indicating noticeable tissue damage only after 24 hours. At the 1 week post-treatment mark, almost all dips were observed to cause tissue mortality and intense bleaching, while only the base revive treatment showing minimal visual damage. For the PEV species at 24 hours, there was limited damage done, and the results remained fairly consistent at the 1 week post-treatment mark. But as circled here, 3 treatments, which were the doubled hydrogen peroxide, revive, and iodine dips, showed significant increase in tissue damage to the fragments. Lastly, for the PLO fragments, at 24 hours, most treatment damage results remained low, with the exception of hydrogen peroxide, but after 1 week, results remained fairly consistent. So looking at all the data together, the doubled concentrations of Lugol's iodine and revive caused damage, with both treatments ranking above a 3 for the great star and cauliflower coral species. The combination treatment damaged the lobe and great star coral, with about 10% of tissue being damaged on the lobe coral fragments and above 30% tissue damage on the great star coral only after 24 hours. And finally, the doubled concentrations of hydrogen peroxide caused the most harm to the great star and cauliflower corals, which both ranked above 3 on the visual scale. Pretty evidently, it's seen that the great star coral was heavily affected with all treatments, backing up past studies that have shown that the great star coral is significantly weaker when compared to other species, such as the lobe coral, which was minimally impacted by all treatments. So with graph, ImageJ, and test confirmation, The doubled concentration data disproved my hypothesis that those two solutions would cause minimal damage to all coral skeletons. Secondly, the combinations of iodine and revive showed damage to 2 species, which again not supported my hypothesis that the combination of treatments would cause the least amount of damage. And then finally, and pretty clearly, both double and base hydrogen peroxide treatments cause very significant damage to all species, supporting my final hypothesis that both peroxide treatments would cause the most damage. So within the testing and data analysis process, I did run into a couple limitations. For the PELO species, I am lacking some data points, which makes drawing a fully confident conclusion difficult, but we are still in the process of gathering them. Secondly, cutting and handling these coral fragments does put some stress on the edges, which could have contributed to the measuring damage done in the image J analysis. We also had the great star coral species act a bit unnaturally, seeing as it died with just being treated with seawater, which was a little bit odd because that was the same seawater it came from, so it would have just kind of died out in the reef. So it possibly indicates that there was another pressing reason for death. And finally, when operating with image J for my purposes, I was calculating damaged tissue based on discoloration, and due to natural coral formation, bumps on the skeleton may have casted shadows or exposed areas to greater levels of light, which could have caused my data to be skewed a little bit. So moving forward, this data will be given back to the Hawaii Nursery to assist in their efforts towards greater coral restoration in the Oahu region. Firstly, this project can be expanded at the nursery specifically by taking samples from separate locations to test the quality of different reefs around the island. Secondly, future experiments can change the length of time that the fragments are kept in each solution to see if longer or shorter treatments yield different results. And additionally, this project can be expanded to other locations, such as in Florida, which they have a sister nursery there, which can expand the knowledge that we know on coral populations to diversify the data. So these are my citations. And then first I want to thank Mr. DeGroat and Ms. Nuno for giving me this opportunity to be part of the program and guiding us through these past 4 years. Secondly, I want to thank my mentors, Christina and Nicolo, for allowing me into their facility and helping me through the experimentation process. Next, I'd like to thank my family and my aunt who stayed with me in Hawaii for an additional week. She will tell you that it was quite awful for her, but that is a lie. She really enjoyed it. And I do want to thank my friends who had to listen to all of my image J complaints. Thank you for just putting up with that. That was really awful. And lastly, I'd like to thank the Marine Chem class of 2026. You guys have really made this a transformative past 4 years for me. So yeah, thank you. Okay, if anyone asks questions, I might not see you, so I apologize. Yes?
First off, great presentation. Second, what is the motivation behind doing these types of tests or doing these types of treatments for these nurseries, these coral nurseries?
So as I kind of talked about before with the, she kind of asked what was the motivation behind doing these treatments to corals as they come into the nursery. So the answer to that is with the increasing in temperatures around the globe, corals out in the wild, once they become bleached, they are more susceptible to pests getting into them. So basically what the coral nurseries are doing are they take the samples of corals and bring them back to the nursery, and through these dips they are able to clean out the pests and disease that may be on the coral, and once they are able to fully grow back to a state where they can replant them, then it will be a healthy coral that can help regenerate the reef that they came from originally. Oh, yeah. So the question was if any of the corals that I tested on have gone back to the water. So my answer to that is in the corals used in Hawaii, they take about 2 years to grow fully before they can be replanted. So some of the fragments that I tested, they will have to take up to 2 years before they can put them back out into where they came from. So at the moment, no, but they could potentially in a couple years.
You plan to be there in 2028?
If they invited me back, I would love to go, but I'm not sure if that would happen.